Thank you. It's a great pleasure for me to be here with you today and introduce a little bit what Accelix is and mainly the the presentations will be run by Chase on the Accelix on the Excelos data and use cases of the Accelix platform. So I am Felix Montero Julian. I am the VP of Scientific Affairs at Accelix here in San Jose. And we prepared this webinar with Excelos together, and this is the agenda that we will cover during the next 58 minutes. So give you some rule of engagements during the webinar, introducing of the speakers. I will present very few slides about Accelix, and then Chase will present the insights into rapid flow cytometry for GMP manufacturing. And the goal is to have a Q&A session at the end. So we encourage you to use the chat also to write the questions you may have during the presentation so we can address it at the end of the webinar. So rule of engagement for today, it's always good to remember. So please keep your micro mute unless speaking, and feel free to turn on your camera if you are comfortable. You can post your questions anytime in the chat, and we will address them during or at the end of the session, mainly at the end of the session. So the session is being recorded as it was mentioned, and the slides or materials will be available after the webinar. And let's keep the discussion respectful and open. So the presenters today is my pleasure to introduce Chase Mockton, who is analytical development scientist at Excelos. And also we'll be joined online by Anika Vijay Kumar, who is a technical lead MSAT in Excelos, who have also extensive experience using Excelix. As mentioned, I am the VP of Scientific Affairs in Excelix. So let's go and move into the into the subject today. So cell therapy is not only the manufacturing part, right? The manufacture of the products, because cell therapy starts from the collection to infusion. And this is the total value chain that we have considering today to discuss, and particularly the collection part and the manufacturing part, because there is different steps that are needed to be under control in order to get the right products with the right efficacy infusions into the patients, starting from the collection of the blood samples, transported or used fresh, infusion of the process they are setting up, the cryopreservation, that is a critical step before sometimes going into manufacturing, and then the manufacturing, handle these samples, cryopreserve, ship it to the lab or to the hospitals, and then infuse to the patients. If we do a very quick overview on the manufacturing workflow, and this is the illustrated here in this cartoon. So the control of these materials is not only at the beginning, but it's also during the incoming, for the incoming quality controls, the in-process, so during the T-cell activation, selection and activation, looking at the transduction for lentivirus and the cell expansion until the final release. But the process really starts at the collection point, because the cellular starting material apheresis is extremely important to be characterized at the point of collection before going into the preservation and storage, to monitor if something happens during the transportation or something happens during the manipulation of these samples. So the connecting of the cellular starting materials with manufacturing is made a success of all these therapies. And we believe that the path of quality starting materials is for the allogeneic cell therapy starts from the healthy donor screening, the pre-collection counting, and then the collection. And we need to, we believe that we need to demonstrate leukopath composition at the point of collection. And the standardizing results across multiple collection sites is also critical in these cases. And also differentiate as a premium suppliers and natural cell therapy partners, maybe also a competitive advantage in front of different other suppliers. So flow cytometry is among the most frequently used quality control tools in cell and gene therapy development and manufacturing, because they provide comprehensive cellular characterization in terms of phenotype definition, heterogeneity mapping, multiparametric analysis, but also to run in process controls, manufacturing consistency, providing real-time feedback in some cases, like what we are trying to do with Accelix platform, but also perform some comparability of batch to batch. Some of these potency and functional readouts can be also performed using flow cytometry, but also flow cytometry is really embedded into the regulatory and quality release requirements to do the identity testing, purity testing, safety assessment, but also, as I mentioned, characterization of primary source of cellular materials. However, flow is a wonderful technology and has been mainly designed around R&D and clinical application. For those that has been working with regular flow cytometers, those are pretty large platforms. We need to have a dedicated QC space, and they have sometimes cumbersome workflows. And importantly, also in the context of cell therapy, we need to have high skill operators to run the assay and to analyze the data. So those assays are sometimes long and expensive. And to do that, so for an industrial application, what we need is to have a platform that can be going to the GMP manufacturing, allows to do the testing at the collection point for the blood markets, but also addressing cell and gene therapy markets. So the main users here is multiple testing points, in-process testing. We can have the placement in clean rooms, have low-skilled technicians running the assay and interpreting the data. And most important here is providing go, no-go data decisions at some point. So Axelis has developed a rapid, fully automated sample to analyze a cell phenotyping platform. So bringing a simple standardized quality control solution to the point of need. How we do that? So with the platform, we provide rapid results without a flow cytometry expertise at any collection sites. We reduce testing workflows time by more than 80% compared with traditional flows, and we increase reproducibility by reducing manual steps and providing, we believe, true sample phenotype by eliminating Western steps or other different shipping conditions that are needed sometimes to run this assay in regular flows. And last but not least, provide cost savings with these accurate automated QC workflows. So the Axelis automated cell phenotyping platform resides in three different pillars. The first one is a cartridge in which the run has been, the sample will be run and analyzed. And this cartridge contains all the microfluidic systems that are needed to run the sample, and also including there the flow cell that is needed to analyze the samples. But also we have reagents that are dry reagent down reagents that has been dry in order to increase the stability, and you don't need to use any cold rooms to keep it storage. These are single-use disposables, and we have a standard and custom assay. The second part is the instrument. That is really mainly an optical and mechanical system. It's a benchtop flow cytometer. They provide 30-minute sample to answer. It's fanless, so it's a clean room compatible. And we have incorporated two different cameras in the system, one to control the fluidics of the cartridge and the second one for the laser verification. And another important component of the Axelis automated cell phenotyping platform is the software, because we developed specific algorithms for these off-the-shelf assays that allows to do the auto classification of the different cell populations automatically and display all the results on the screen. The full data can be exported like FCS files for secondary analysis in the cases needed, and also enables the 21 CFR per 11 compliance. Today, we have Axelis assays for the phenotyping and enumeration, so TBNK16 lysis, TBNK16 no lysis, T cell lysis, T cell no lysis, stem cell lysis. And we have here the populations that are detected and the sample types that we have tested so far. I mean, the most important one in the context of the cell therapy and cell collection, the apheresis for TBNK lysis, PBNCs for no lysis, apheresis for T cell lysis, PBNCs, and also mobilized apheresis in the case of the stem cells. Very quickly, a technical point here. So the flow cytometer that we are commercializing here is the multicolor spectral analysis, and basically don't have any very complex setup that needs to be happens. So it's a 6-color panel with single blue laser. It's a stable fluorescent and forward scatter detector. The detection range goes 510 to 800 nanometers, no calibration compensations needed, no gain adjustment required, and we have an automatic spectral mixing algorithms inside that allows us to analyze the data in the proper way. There is no complex filter train and it's pretty low equipment cost. The workflow is at two steps for the immunophenotyping of T cells and lymphocyte subset. So you have the cellular material, tow and go with no wash, added to the dry down reagents here, 40 microliters of the sample, incubate for 2 minutes, and then incubate 20 microliters of these of the dry down reagents into the cartridge, and then we place the cartridge in the instrument for 30 minutes staining, acquisition, and automated analysis. So what happens after the loading of the cartridge? There is no more manual interventions that can reduce the variability linked to the testing. In this case, there is a 15 minutes incubation in the cartridge. The sample is diluted with some buffering included into the cartridge, and if needed, some differential red blood cell lysis. Then the sample is pushed into the flow cell. The sample is excited by the 488 nanometers laser diode in the flow cell, and the signals are captured using a silicon multiplier array to detect the fluorescence. Then the system do the spectral mixing, so the compensation for spectral spillover is applied to all the data, and the FCS files containing the spectral mixing data are generated. So the assay-specific algorithms that identify and classify cell subsets, and they provide some pseudo-color-coded scatter plots that are included into the PDF for quick visual review of the data if needed. So why is this applying objective logic via automated analysis minimize measure uncertainty here? And this is only to illustrate at the right-hand side of the slide how this ambiguity sometimes in the threshold plasmids due to the noisy or poorly defined borders between the cell populations leads to inherent subjectivity, and increased measurement uncertainty. Here you see the mean values obtained with 3 different analyses in which we asking to place the regions, and you see A, B, C, the thresholds, and you see the main difference we can obtain. So the use of these auto-classification algorithms sequentially set thresholds based on predefined event density parameters, identify properly the cell subsets in the samples. But we have also included some warning flags for example, low events acquired, unexpected noise, or some out of tolerance cell frequencies that ensures data integrity also. And as I mentioned before, FCS files are exported with every run, and in the case the runs needs to be run doing some data analysis, some manual analysis using a third-party software. So how to better control starting materials at the point of collection. And this is the case, for example, for the leukopacs, in which at the blood collection, the blood is collected, a sample is sent to the outside lab. They can wait for 8 to 48 hours after the delivering the results, and only after that the samples are shipped to the commercial customers. But with Accelix, we believe that the blood collection plus the point of the testing at the point of collections make this availability of samples more quickly to the commercial customers needed. And if you want to learn more about the leukopacs in general, how these leukopacs are impacting the cell therapy and donor screening happens with Accelix, I invite you to look at these very nice tutorials we have on the website from our leukopac lady, Jennifer Chen. And with that, I think that the starting material quality defines the destiny of every cell and gene therapy products. And having that, having these controls will make better products in the future for our patients. Now I will over to you, just to present the experience of Excelux to the BCA community. Thank you.
Wonderful. Thank you for that great overview into the Accelix platform and the kind introduction. So yes, I'm Chase. I've been with Excellos for 3 years, and I'm prepared this presentation alongside my colleague Aniha. And traditionally, we don't have, flow cytometry training, but we've really found that the Accelix is simple to use and has really elevated our manufacturing workflows. And I hope to demonstrate that through some of the data that we've collected here today. So to give a brief background on Excellos, we are originating from the San Diego Blood Bank that serves the greater SoCal area, and we've been incubating here since 2012. And the San Diego Blood Bank has a rich history, over 70 years of donor management and blood banking expertise, and their mission is really core to blood banks everywhere. But what we believe is in leveraging the donor-derived insights into therapeutic impact. And that's kind of where we spun out into Excellos. We are a contract development and manufacturing organization. Since 2021, we've been independent, but we've brought on a core group of 18 seasoned members that have that history in blood banking, donor sourcing, and we've developed these GMP operations at our new facility here since 2023. And we've moved into this multi-purpose commercial use facility. We built out 5 different clean rooms that were able to support different manufacturing of GMP programs from clinical to commercial. And we've really maintained, we're only 2 miles away from the San Diego Blood Bank, and we maintain this key relationship, not just with them, but building it with other blood bank centers of excellence. And as a comprehensive CDMO service, we kind of start with our traditional blood banking roots and providing donor cell collections. And as I'll show throughout this presentation, we really believe that upfront characterization is necessary and impactful towards creating end-stage drug products that make a difference and have clinical relevance in supporting complete responses, partial responses. So from donor collection, we also offer a cohort of purified cells that we provide as isolates that you can kind of see up here in the upper right image. And these are non-clinical or research use only products that we do offer allogeneic off the shelf. These range from CD3 cells, NK cells. We also do hematopoietic CD34 plus cells, and we do use the Acellix as a quality control release metric to ensure that when we perform these isolations, we have the right level of purity to make this an impactful product. And then the bulk of our organization really is focused on kind of this full tier service from both process and analytical development scope. This allows tech transfer. We do scaling of different processes for CAR-Ts or other types of cell therapies. And we also are really invested in supporting both clinical phase 1 through 3 trials, as well as commercial manufacturing for GMP materials. And of course, we have the storage, shipping, logistics, and everything that comes from getting it from the manufacturing facility to the clinics where it can go ahead and impact the patient. And we're really always thinking about the patient here in our design of the different manufacturing workflows or logistical operations. So with that said, we wanted to focus today on some topics within allogeneic cell therapies. And to start off, I think it's always important to understand the different challenges that we face. The first one being the complex tumor microenvironment. This is really inherent to why we're seeing some resistance to different modes of therapy, and especially these cell therapies, and worth understanding this inherent resistance. We believe that there's an inability to select the right cells. This could be as simple as when you're considering autologous therapies, you have patients who have received chemotherapies, which can have drastic effect on their native immune cells, that when we pull those blood draws, leukopaks, and try to perform manufacturing, they're not necessarily the right cells. Or kind of on the other side of allogeneic therapy, even though we're able to select T cells, we find that in the literature and in our experience, there's even subtypes of T cells, such as, stem cell-like memory, that is more the right cell that we need. So these are important considerations. Something else worth mentioning here, which I think really plays into our strategic partnership with blood banks, is that a lot of our clients are always coming to help them logistical power lift of providing that starting material, but not just any starting material. They're trying to find high quality starting materials that they can use that are going to be lead to the highest level of success in their individual workflows. And that's because, kind of to summarize it here, even though we're striving to create these identical manufacturing processes and really controlling everything through critical process parameters and getting that drug product that's quality control released against acceptance criteria, ultimately we're starting with a variable input. We have that donor-to-donor variability up front. We found that this is likely the largest contributor to variability in the drug product and the clinical responses that we're seeing. So with our model, we're really trying to understand this donor to dose aspect. With the donor, we want to do more strategic sourcing, but what does that mean? That really means that we're creating this partnership and we're better characterizing that starting material. Ultimately, this can be either creating critical material attributes within item specs, so we have a go, no-go decision for whether or not that leukopak or cord blood will be used downstream, or smart material selection to identify high-quality donors for specific sponsored programs. And we think that this upfront characterization will ultimately impact the dose. And our goal really is to create more safe and efficacious doses of that drug product. Defining the critical material attributes is important. And going beyond just, characterization, we don't want to just characterize everything. We're actually trying to look at what we believe or what the literature has shown correlates with the clinical response. So you are likely familiar with the leukopak products shown here, and this really is the majority of what we work with in terms of the starting material. And it's excellent because it gives us that high concentration of white blood cells that we're typically targeting for downstream selection and additional manufacturing. But when we go back into kind of some of our internal databases we have around this, we're able to show this phenomenon where based on the donation age, for example, we're seeing differences in the immunophenotype where increasing age, we see an increase in the CD4 populations and a decrease in the CD8 populations. And as I'll show in a subsequent slide, we do believe that this type of ratio has been shown in the clinic to impact manufacturing processes. So this is an example of where, the cell repertoire is different, not just from donor-to-donor variability, but also with demographic inputs such as age. And on the other side, what does this donor-to-donor variability mean when it comes to the cell repertoire? So I'm specifically showing a individual case study where they were trying to isolate PBMCs and expand out a niche cell type, the gamma delta T cell, and they're showing that the populations are represented on the y-axis on day 12. So this is a typical proliferation, isolation and expansion protocol where you're seeing the PBMCs isolated on day 0. It is exposed to Allogeneic, which is a specific antagonist of gamma delta T cells, as well as IL-2 to support proliferation. A lot of these processes will add in an intermediate step. Here on day 6, you can see that they're doing an alpha beta T cell depletion to remove any impurities from the alpha beta T cells. And on day 12, we finally have our functional characterization of that N gamma delta T cell product, as well as cryopreservation for downstream infusions or additional processing. But the reason I wanted to show this here today is really to highlight this phenomenon where this individual study observed a range from 10% to 40% of natural killer cells across the different leukopaks that they were starting with on day 0, but they found a negative correlation with the percentage increase of gamma delta T cells on day 12, such that in hindsight, or what we would use with this type of information during development, would be to impose a critical material attribute at approximately 25%. To say that any leukopak with greater than 25% natural killer cells, we either have to partially deplete them or we would not recommend these to move forward with manufacturing because we're not going to get that ideal drug product at the right yield or purity that we require. So what we've done here is we've actually shown that, a solution could be to provide that characterized leukopak material up front. So Ania and I set up a workflow in which we did just that. We screened for the leukopaks. First, I just want to take a second to show why we picked the Accelix technology to do this, and it will emulate some of what Felix had previously said, but as an end user, we feel it's important to repeat. The simplified instrumentation is actually really important. On the left, you'll see an example of a traditional flow cytometer, very big and bulky, requires extra computers and softwares for all the downstream analysis, and ultimately takes up a lot of space. And as a CDMO, we really care about our benchtop space that we have because we have to have so many different instruments to provide to a variety of clients. Astellix on the right here has a really small footprint, and it's that cartridge-based technology. We also went ahead and did this exercise where we predicted how long it takes to actually train an individual analyst and operator. Traditional flow cytometry, assuming they have some level of baseline manufacturing and flow cytometry understanding, takes about 30 hours, and that's with classroom training, supervised runs, independent runs, and any additional troubleshooting. In our experience, it only takes about 3 hours to perform this on the Astellix instrument, meaning it's a lot quicker to train and less time that we have to invest in that upfront training of analysts and operators. And I'm also excited to share that we pulled together what it would look like to do a similar T-cell characterization on traditional versus Astellix flow cytometers. And it's a great reduction in materials. We just need the T-cell kit and the QC kit for GMP stage with some micropipettes to be able to load these cartridges for the Astellix technology. And having worked with several tech transfers into a GMP state, I can tell that this would cause me a headache given how many different materials need to have individual item specs, sourcing, quality assurance oversight for release, and materials management within our laboratory. So Astellix greatly reduces that and makes it a lot more feasible. And lastly, that time to results, I think, is something that's really important to understand here with traditional flow, performing all of the immunostaining, performing the data acquisition, and ultimately doing any type of compensation analysis offline on the different software provided does take about 3 hours, we kind of imagine, in a controlled workflow with a trained operator or analyst. Astellix, however, delivers 3 times faster results, given that it's just a similar sample prep and the data acquisition is all automated. So this is our rationale for choosing the Astellix, but what we've been able to do is a proof of concept point of collection. That is when we receive the leukopacks, we can pull a sample and process it on the Astellix using both the T-cell and TBNK kits. The data here kind of demonstrate what that characterized leukopack may look like. So the T-cell subsite ratios on the left shows the CD4 to CD8 populations. And one of the reasons that we believe it's valuable to have this up front is because the literature demonstrates that low CD4 to CD8 ratios has an increased risk with manufacturing failures. The reason being is we find within proliferation conditions, the CD4 cells are actually important for supporting CD8 ratios proliferation, and that CD8 cell type is ultimately the effector cell. And so any non-ideal ratio may impose a manufacturing failure. The graph on the right, we're kind of showing, taking a step back and looking at the different subtypes. So we have CD4 percentages, we're already CD45 percentages, CD3 plus populations, CD14 are our monocytes, as well as the CD19. So 2 quick stories on why we believe this type of data set upfront is valuable. One, the literature shows that increased level of monocytes can actually inhibit CAR T processes, whether they're inhibiting the T cell function specifically due to states of stress or inflammation, or they're performing phagocytosis of different viral transduction tools, causing less efficiency when it comes to transduction for a CAR T process. And then similarly for the CD19 cells, we have had clients come before and say that they have a lower limit specification for their leukopacks when it comes to CD19. And while I won't give those specifics, I'll just give a general example here to show that hypothetically, if that lower limit was 35%, we would have 3 leukopacks which would not meet the specification. And up front, we can make that decisions where it doesn't get discarded, but as a CDMO, we can divert it to other purposes. And when we kind of step back at the field of CAR T manufacturing, for example, where we know it's very expensive to manufacture a single dose, anywhere up to $500,000, Eliminating these three leukopaks up front is really important because it's going to be cost savings at the end of the day, which impacts not just the developers, but also make sure that the patients are getting the treatments that they need. In our hands, I'm going to show two scenarios where we, in hindsight, want to demonstrate how valuable that characterized leukopak information would be based on what we've learned through some of these manufacturing programs. The starting material specifications on the left is something that we think needs to be implemented more and more, whether it's what we're going to show here for our RUO manufacturing or for more larger GMP manufacturing programs. But specifically, and probably rather intuitively, we're showing that an increase in the CD3 plus population in leukopaks does lead to an increase in yield per run in our hands when building a research use only CD3 plus T cell isolate. And it is intuitive, but at some point I put on my business cap and I realized that if I were to keep this program or how to make this program better, well, we would want to impose a lower limit of approximately 45%. And so in hindsight, that's what we've observed, and that's what we're doing more moving forward to making sure we're getting the best return on investment with these individual manufacturing processes. On the right here, I wanted to transition to a little bit more specifics on a CAR T cell process, something that Ania actually was developing using the CliniMax Prodigy. And it was for autologous, but it can also be translated easily for allogeneic therapies as well. But this screen is a specific step in the manufacturing process in which the CliniMax Prodigy actually requires the operator to input characterized leukopak information. And while we're showing here that the Accelix can be a great tool in which we're getting this nicely auto-classified output information that can go into the Prodigy software to proceed with the manufacturing, one thing to keep in mind is that this assumes traditional flow. So it does give us a 3-hour hold window while it's expecting the operator to pull a sample, pass it out of the clean room to the QC team, and await their 3-hour process to acquire this type of data to bring back into the clean room. Now, we appreciate that the Accelix technology is able to decrease this time again about 3-fold so we get faster information. But we also, as a CDMO, believe that it would be even better to have this information at point of collection and upfront provided on a certificate of analysis so that we can take that 3-hour window not just down to 1 hour, but actually eliminate it, because when we're in the clean rooms, we have our operators, which we don't want to be idle. We have our EM technicians continuously monitoring the process, even if they're waiting for this information. And we don't want to risk any errors on the QC side, which could cause delays. And this all is really about having that in-process parameters up front to reduce any risk of failure. So how does this ties in with the Accelix 360? So our kind of goal as an organization is really to reduce that endpoint variability in the drug product to make sure that the patients are getting a more consistent product. We do this with the upfront characterization of the starting materials. And in our hands, I've kind of shown how we've done this with both using the Accelix for immune profile screening. We actually do lean a lot on the demographic information in some of our E360 algorithms. And I quickly want to segue as well into some of the tools we use both to assess the metabolic fitness as well as the effector function potential of the starting material. And we believe that this cohort of information provides better prediction of which therapies or which drug products are going to be more efficacious to use. It's also leans itself to understanding the manufacturing process. And this is all pretty much done by quality by design assessments in different manufacturing scenarios. So beyond the cell repertoire immunophenotyping analysis, we've also found this measurement of polyfunctional strength to really correlate with the clinical response as shown here. This is done by analyzing the starting material, and the specific technology evaluates 32 cytokines at a single cell level for the individual donation or leukopak material. It characterizes that single cell for having 1 two or more different groups, such as inflammatory or effector function, and uses a proprietary algorithm to create this polyfunctional strength index score. In the clinic, as shown here in this literature report, we see that the higher polyfunctional strength index actually is correlated with more response of the patients to these cell therapy treatments. So in our hands, in addition to this Excelix, we're actually performing this donor variability assessment for polyfunctionality. And you can see that across 7 donations, we have a range of different polyfunctionality scores. And what this means, at least in terms of potency, we don't quite yet have clinical data, but potency is an important quality control aspect, and a higher polyfunctionality score, such as donor 2 here, shown in green, correlates with better cell killing. This is a higher E score, and the yellow, lower polyfunctionality donor 4 actually correlates with more nuanced or less appreciable cell killing. Again, on the y-axis, I know it's small here, but we're actually looking at an increase in the y-axis, is higher level of cell killing. These are different effector to target ratios, and we really get much better response in this high-scoring donor when characterizing the PBMC. We start to see a drop-off when we have lower effector to target ratios for a low-performing donor in this effector function analysis. And this really lends into our vision for creating these successful GMP drug programs. We want to move into this space where we're gaining a lot more insight with more deeper characterization upfront, beyond just viability or cell number, total nucleated cells. And this is really where we're incorporating our E360 platform to utilize the demographics, the cell repertoire, which ideally would be collected at point of collection for leukopex or other starting materials, as well as these more advanced screening tools, metabolic health, looking at the starting materials ability to go from glycolysis, oxidative phosphorylation to glycolysis, as well as this polyfunctional score. This information will be better for insights into the downstream drug product, both yield and quality, and ultimately our goal is to make sure that what we're performing in starting material characterization and managing these manufacturing workflows will lead to clinical alignment with response. And I do think that this is more of a strategic partnership. We really rely on the experts in blood banking, such as yourself, in determining how best to provide and characterize this starting materials. We are grateful for the teams at Excelix for creating this technology that gives us reliable and rapid characterization ability using their flow cytometry instrument. And we're continuing to do our best here as a manufacturing center of excellence in which we're understanding the needs of our sponsored programs, and it's really the strategic partnership that's going to allow us to make a difference in the future. And I want to segue again just to show another application of the flow cytometry that we found very advantageous. And this is for our off-the-shelf research use only CD34 plus or hematopoietic stem cells derived from cord blood units. So Aniha really led this process, but it is a approximately 3-hour immunomagnetic isolation of these hematopoietic stem cells. Really important, though, is our ability during development to get rapid insights, given how quickly we can get the information back from the Excelix instruments. And this allows us to export the files, and we're able to see here, when optimizing the variables, what leads to the highest level of purity and or yield for our CD34 plus cells. As shown here, this is just a demonstrative data where we're meeting our CD45 plus lower limit of 95 or 90%, and we're achieving 94.5% positive cells. And of those, we get 96.8% positive CD34 cells. So it's rapid feedback that aids in development. But the other aspect is the minimal sample usage. With traditional flow, we require not just the stained sample, but single stain controls, fluorescence -1 controls, unstained controls. And with this niche cell population for CD34 cells, given that they're already such a low frequency in the cord blood units, we're really adverse to taking so much of our final product in terms of just quality control, release testing, and screening for purity. The Accelix is a huge advantage. We only need 40 microliters or 40,000 cells to perform this. Again, thinking about the logistics and the business side of this, I've demonstrated our historical data sets in terms of yield for CD34 plus cells using a histogram. And I really want to focus on the leftmost bin in which we've yielded less than 0.5 million cells. 5% of this historical lot that we manufactured did not even have sufficient product for vialing. 15% we've calculated here on the leftmost bin had a net loss on return of investment, even if we were able to vial 1 or 2 at the lower concentrations. Why? We didn't really have a lot of insight into how many CD34 plus cells were actually present. Which is something that we think in an ideal situation we'd have upon receiving these to inform our manufacturing decisions. And while we have Accelix's in-house, it's also critical to keep in mind of these logistic workflows. We have a very short window in which we can go from collection to blood processing at the blood banks to receipt here at Accelix in terms of starting the manufacturing process, which means even 1 hour can make or break whether or not we're able to proceed, and thus we don't always have enough time to perform that characterization or immunoprofiling. If it was able to be done before and we received this information, it would be very advantageous. So again, that's the ideal solution to receive the CD34 plus concentration screen on the CBUs. But in the interim, we've been able to at least create an item specification in which we have imposed a minimum TNC count. We pulled out the last 20 lots that we manufactured, and with this interim solution, we have been able to have good success. We've had over 300 runs performed. Here you'll see that donation 8, we've actually had a manufacturing failure where we did not quite meet the purity requirements, and that's okay. And we actually wanted to show this here to demonstrate the last kind of critical component of using Accelix as it is for this quality control release parameters. And this information is important to make sure we have the highest quality of products going out to our clients. So to summarize briefly, we believe that the Accelix instrumentation as a rapid flow cytometer is very advantageous to use at various points in the manufacturing workflow in our hands and based on our data sets. We're seeing more and more, and we believe that development work will increase the amount of critical material attributes, not just on the target cell types, but also on impurities, and that is going to better fine-tune our manufacturing processes. But this can only be accomplished if we have that starting material screened up front. We showed with the CliniMax Prodigy that more automated closed systems for processing are going to require these inputs for CD3 plus populations, for example. It could also be used for in-process monitoring of that CD4 to CD8 ratio or other types of cells that may impact that specific manufacturing workflow. And lastly, we believe that it is advantageous for quality control release testing, shown here with our release testing for our natural killer cell research use only cell isolate offering. And really kind of just going back to why we selected Accelix, it's very easy to train analysts. We get this rapid results, and we believe that it's advantageous for these reasons. And a lot of this was done more in the development or RUO space, but ultimately this has such huge impacts for being able to really minimize the amount of training and work needed to get a really good instrumentation for these GMP drug product release workflows. So with that, I want to thank you for your time and